Inorganic compounds are compounds that are usually missing the element carbon. There are a few exceptions. CO2 is an inorganic compound that does contain the element carbon. The most important inorganic compound is water. Water is the most abundant compound found on the planet. Water has many unique properties, one of which is when water freezes, water expands, thus becoming less dense and floating. This allows marine life to survive. If water did not become less dense when it froze, ice would sink to the bottom, thus crushing marine life. When ice melts, the hydrogen bonds, which hold water together, are broken. Water is a polar molecule, which means it has a partial positive region around the hydrogen atoms and a partial negative region around the oxygen atoms. This allows water to take on two main properties: cohesion, when water loves itself or is attracted to other molecules of water, and adhesion, when water is attracted to other things. Cohesion can result in surface tension, and can also result in high specific heat of water. Adhesion, water loving other substances, allows for capillary action to take place. Capillary action is when water, absorbed through the roots, flows up against gravity along the xylem and the phloem of a tree, thus giving water and transporting water to the leaves. pH measures the amount of acid or base in a substance. Acids produce H plus ions in solution, and bases produce OH minus ions in solution. Acid is from a pH of 1 to 6.9, and basic environment is from a pH of 7.1 to 14. Strong acids are from 1 to 1.13. Strong base from about 11 to 14. Neutral is in the middle. Water is an example of a substance that is neutral. Buffers are substances that prevent a sudden change in the pH. This helps maintain homeostasis in the organism. Mixtures are where two or more substances are physically combined. There are two types of mixtures that we talked about: solutions and suspensions. An example of a solution would be salt in water. An example of a suspension would be sand in water. The main difference between a solution and a suspension is in a solution, the solvent dissolves the solute, and the solute gets dissolved. In a suspension, the solute is not dissolved. It separates or falls out of solution. For example, sand and water, if you left it alone, the sand would fall to the bottom or separate to the bottom, and the water would rise to the top. Also remember metrics. Make sure you guys remember the basic unit of length is meters, basic unit for temperature is degrees Celsius, basic unit for mass is grams, volume of a liquid is measured in liters, and volume of solid is measured usually in cubic centimeters. Your prefixes: kilo, hecta, deca, base, deci, centi, milli. Kevin hates dates because dates cost money. Kilo is 1,000, hecta is 100, deca 10, base 1, deci 0.1, centi 0.01, milli 0.001. Scientific method. Remember the difference between observation and inference. Observation is something where you are using one of your 5 senses or many of your 5 senses, such as seeing birds flying into a birdhouse. An inference would be a logical interpretation to your observation. So if I watch the birds fly into the birdhouse, an inference could be that birds are hungry and they're feeding. Another inference might be that the birds are hiding from a prey or a predator. Another inference could be that the birds are trying to get shelter from a storm. Hypothesis must be able to be tested, and then an if-then statement. For your experimental groups, remember the control group is a group where there are no changes that are made, that are left alone as a reference or a comparison. Your variable group or experimental group has an independent variable and a dependent variable. Independent variable is the variable that is changed, or the variable that we are testing, and is usually on the x-axis. And the dependent variable is the variable where we are measuring, or the variable what is called responding, and is on the y-axis. Of the macromolecules that we talked about, carbs, lipids, proteins, and nucleic acids, all of them contain the element carbon. Carbon is a unique element in that it can form or bond to 4 other elements, including itself, and form what we call a carbon skeleton. This allows organic molecules to become very large in their molecular makeup. Carbohydrates contain the elements carbon, hydrogen, and oxygen in a ratio of 1:2:1. They are also called sugars, and usually end in the letters OSE. The 2 main functions of carbs are the primary source of energy for most living things, and are also involved in cell structure. There are 3 types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides provide quick energy and are referred to as simple sugars. Polysaccharides are referred to as complex sugars and provide long energy. These are the 2 main types of carbs. Disaccharides are made up of 2 simple sugars. An example would be lactose, which is found in milks and is often connected to lactose intolerant. People have the inability to break down this disaccharide lactose. Examples of monosaccharides or simple sugars would be glucose, which is also referred to as blood sugar, and which provides energy and travels through your blood to your cells, and fructose, which is often found in certain fruits. Both of these, again, are simple sugars and will provide quick energy. Polysaccharides are complex sugars, again, provide long energy. Some examples of polysaccharides would be glycogen, which is found in the liver of humans. When glucose travels through the blood with the help of insulin to the liver and is converted to the complex carb glycogen. Starch is plant sugar and is referred to as stored energy for plants. Cellulose is also considered to be a complex carb, but is not used for energy. Rather, it is used for structure. And the last polysaccharide is chitin, which is found in the exoskeleton of certain insects. Again, monosaccharides, disaccharides, polysaccharides are all types of carbs. Monosaccharides providing quick energy, polysaccharides longer energy. The monomers, or the subunits of a polysaccharide, are monosaccharides. Lipids are another organic compound, contain the same three elements as a carb: carbon, hydrogen, and oxygen. However, there is no ratio of 1:2 to 1. In fact, you will find more carbons and hydrogens than you would oxygens. Lipids are made up of monomers or subunits: the glycerol molecule and 3 fatty acids. There are many functions of the lipids, reserve energy being their primary. Once your body breaks down all of your carbs, your body then uses or begins to break down lipids for energy. They're used for cell membranes. Remember the phospholipid. Is a molecule that makes up the cell membrane. Lipids are also involved in insulation, helping to maintain homeostasis, and cushion and support to protect your organs. There are 3 types of lipids: fats, oils, and waxies. There are 2 main types of fats: saturated and then unsaturated. The difference being, saturated fats have no double bonds between the carbon skeleton. Unsaturated fats will have 1 double bond in the carbon skeleton. Polyunsaturated would have more than 1 double bond between the carbon skeletons. An example of saturated fats could be animal meat or animal fat, butter, and cholesterol. Examples of unsaturated fat would be your oils and margarine. The third organic compound are proteins. Proteins are made up of monomers or subunits called amino acids. There are 20 known amino acids. Those 20 amino acids can be broken up into essential amino acids that you would get from your diet, and then non-essential amino acids, which your body makes. The amino acid structure is different than that of the structure of carbs and lipids. It contains 3 groups: the amino group, the carboxyl group, and the R chain. Every amino acid has the same amino group and carboxyl group. What is different is the R chain. So if there are 20 amino acids, you would have 20 different R chains. How these amino acids are arranged can lead to the body making thousands of different of proteins. Because there are thousands of different proteins, there are many functions. We're only going to focus on a few. One of the main functions of a protein is protection. They are involved in creating what are called antibodies, which help fight bacteria and viruses. Proteins also can aid in the transport of certain material, such as hemoglobin, which is found in your blood, and transports oxygen. It's involved in structure, such as muscles. And one of the most important functions of proteins is that it acts as a catalyst, as enzymes. All enzymes are proteins, but not all proteins are enzymes. Enzymes can do one of three things. They can speed a reaction up, thus acting as a catalyst. They can lower the activation energy, thus saving energy, and they can be reused over and over again before they begin to break down. One cell will contain thousands of enzymes, each enzyme being specific to what it does. Enzymes have what is called an active site. That is the part of the enzyme that works on what is called a substrate, much like a lock and a key. For every lock, there's usually one specific key. Enzymes can be affected by temperature and pH. If it is too hot, enzymes can be denatured. When an enzyme is denatured, the active site is destroyed. Cold temperatures usually will slow down the rate of reaction. There are different levels of pH in the body, both acidic and basic, and different enzymes will work in different environments depending upon where they are found. The last organic compound are nucleic acids. There are two main types of nucleic acids, DNA and RNA. DNA is responsible for your genetic information. While RNA is responsible for making proteins. The subunits or monomers of a nucleic acid are called nucleotides. Lastly, remember the two processes that help build these macromolecules from their monomers into their polymers are dehydration synthesis, which is building by removing water. Make your polymer by removing water. And hydrolysis, we are breaking down from a polymer into smaller substances, maybe monomers, by adding water.